I've recently been making videos on how ivermectin or fenbendazole might be used in cancer based on some of the emerging evidence. While these are not standard therapies at all, obviously, and many of you have been commenting that perhaps one of the mechanisms of action is because they target parasites, which of course, these are what these compounds are known for. And some of you in the comments are suggesting that it's precisely because they target parasites, it might help treat against cancer. Now, this idea is not without merit, it's just not widely accepted. And I'll discuss all of this in a moment. My name is Dr. Mikola Rashek of Merogenomics, and let's get started. This concept, in terms of why I should be investigating this, was brought to me by one of the scientists I collaborate with, and he said, look, you should be looking into this idea. And indeed, how these drugs might be helping fight cancer is an angle you might not be expecting. And I'll explain that obviously right away. And this is one of the reasons perhaps why these drugs are potentially underappreciated for their possibility of how they could be helping fight against cancer via targeting parasites. Now, this scientist said, look, leave me out of this. I want to be anonymous, which kind of shows you how ridiculous it is that scientists want to remain anonymous because this concept I'm about to discuss is simply not widely accepted. And I guess you got to protect your reputation, even though from a scientific point of view, it has some merit. All right. So then let's talk about parasites and cancer. Now, we do know that certain parasites can, in fact, cause cancer. So the very first paper I want to talk about today is from 2016. And while the paper is old, the reason why I want to discuss this paper is because it analyzed literature, how parasites might be involved in cancer development all the way from 1970 to middle of 2016. And they came to the conclusion that infectious agents, well, this was already known, can be characterized as carcinogens. And they summarized this for us, and they mentioned, look, at the time of writing, 4 bacterial species were considered carcinogens, meaning they could contribute to development of cancer, 7 viruses, and 3 different parasites. Now let's talk about the parasites that were known at the time. 2 of them were fish-borne liver flukes. I hope I pronounced the name correctly. And they can contribute to the development of, I think it was cholangiocarcinoma. Either way, not sure if I got the name right. It means cancer of the bile ducts. Okay. And the other one was blood flukes, and they can lead to bladder cancer. So this was already well established by then that certain parasites can indeed cause cancer. One more they talked about is parasites that cause malaria. While it was not considered to be carcinogen, it is known that it can help to reactivate Epstein-Barr virus and therefore lead to cancer as well. I think it might have been Burkitt's lymphoma. Can't remember. But either way, that's parasites and cancer. And it's more like, how do parasites contribute to cancer? That was interesting. And it's through chronic inflammation. Excuse me. And chronic inflammation, something that eventually all of us experience, excuse me, as we age. And what happens, chronic inflammation can activate molecular pathways that eventually can elevate the risk of cancer mutation or mutations in your genetic code, which could lead to cancer. Of course, there is also damage to the tissue, and that means immune response and so on. All of this together could be helping cancer development. And the reason why I thought that was interesting is because these are issues that are found often with parasites. So this is not something unusual. But now, in order to understand how parasites might be helping cancer and therefore how fighting parasites might be helping fighting against cancer with such compounds as ivermectin or fenbendazole, we need to talk about regulatory T cells. Now, T cells, brief overview, you can divide T cells into two broad categories. CD8 T cells, those are cytotoxic T cells. Those are the guys that go in, cavalry, and destroy anything that is rogue. They need to recognize it. So something that is broken has to be presented to them. And then they go in and they destroy either rogue cell like cancer cells or infected cell. Now, those are cytotoxic killer T cells. The other ones are CD4, and CD4 T cells are referred to as helper T cells. And there's many different types. So the most famous division of categories is Th1. Th1 helper T cells are involved in fighting internal problems, such as parasites. So they cause inflammation. That's their way of actually fighting. And if you dysregulate those guys, that's where you can end up with autoimmune conditions. Excuse me. And the other ones are Th2. And Th2 are known to fight external problems. So they're the ones that help to activate, for example, B cells. And therefore B cells will produce antibodies to target something external. They also help to activate mast cells. And if you dysregulate those guys, this is how you can end up with allergic reactions, for example. And what's interesting is those two work against each other. So if you're activating Th1 cells, they will suppress activation of Th2 cells and vice versa. And parasites activate the Th1 response. But there's more helper T cells. There is also Th17 T cells. Those help to fight, for example, fungi and certain bacteria. There's follicular T cells. Those happen to help B cells, the ones that produce antibodies, get really precise in how to target the pathogens with the antibodies in germinal centers. That's where the antigen, such as fragment of, say, spike protein, is presented to the B cells, and B cells are like, this is what we have to target. So those are follicular T cells. I described that in detail in one of my past videos, which talked about some really interesting hypotheses there. And then there's also the regulatory T cells, and these are the hero of our story today. So what are regulatory T cells? These are the cells that make sure that there is homeostasis and all this activation does not go out of whack. So they make sure that once the work of the immune system is done, they shut it off so it doesn't continue, doesn't harm us. As a consequence, you can think of regulatory T cells as ones that suppress the immune system. And this is the key to this story. Now, regulatory T cells, besides CD4 protein on their surface, they also will have CD25, will become important for later of the story. And also FOXP3 is another famous marker. But the issue with regulatory T cells is that it's a complicated story because there's many different types of regulatory T cells, and they have many different functions. They have many different protein markers, and it's hella confusing, really. And as a consequence, oftentimes we really ignore that subject, and they also don't make up much of the whole T cell population. A good example is when we study vaccines, T cells, regulatory T cells usually are ignored. It's just simpler that way. Now, how do T cells, regulatory T cells, function when it comes to cancer? And the old school message was that regulatory T cells help cancers because it was observed that when you have buildup of regulatory T cells, either in the blood or in a cancer environment, that helps cancer grow. Why? Because as I mentioned, regulatory T cells will suppress the immune system, and it can suppress the immune system response to such a degree that the immune system is no longer properly fighting cancer. So cancer cells can take advantage of regulatory T cells to evade immune suppression. And this is what is referred to, like how do cancers evade immunity and immune system fighting it? It's through regulatory T cells. Cancer cells can even put out chemicals that will attract regular T cells. Come here, come to my environment. And regulatory T cells are just totally seduced to come in there, and they start working, and they start suppressing the immune system to properly attack cancer. Now, in numerous of my videos, I also talk about how a lot of cancers, if not majority of cancers, have altered metabolism, where they use sugar in a specific manner, where they break down sugar for energy without use of oxygen. This is referred to as Warburg effect. It's well understood that this happens in many, many cancers. And as a byproduct of that, cancers also produce lactate. Now, I mentioned in one of my past videos how lactate can help in metastasis. I also talked about in that video or also another video how cancer cells can store sugar in order to be more aggressive later on. So check that out. And here is another aspect of how cancers can use this altered metabolism to their advantage, because this lactate, it lowers the pH, so it makes its environment more acidic. This is not friendly to the immune cells, and this is toxic to the immune cells. Immune cells that are supposed to be fighting cancer, they can't really use lactate efficiently for food, but regulatory T cells can. They have enzymes that can take lactate and still put it back into mitochondria and burn it with the use of oxygen, proper energy burn. They can thrive in that environment. So you can see how this is of benefit to cancer cells, and regulatory T cells are just fine. Now, that used to be the old story. However, it's also known that regulatory T cells can have this dual role. Not only can they help certain cancers thrive and grow, in other contexts, they can actually attack cancers and make sure that cancers get killed. And those are the type of cancers that thrive very well in highly inflammatory environments. So, for example, cancers that arise because of, say, bacterial infections, like I think maybe colon cancers was mentioned in one of the articles I studied. Well, here comes those suppressing regulatory T cells that will suppress the immune system, and boom, that inflammation that was helping cancer, that particular type of cancer that was thriving in an inflammatory environment, it is now suppressed. The inflammatory environment is removed thanks to regulatory T cells, and cancer has no chance. So you see, there's this dual role of these regulatory T cells. But everything I just told you about regulatory T cells and cancer can also be said about parasites. So everything I just said, you could literally remove the word cancer and insert the word parasite, and the same story emerges. So parasites also help to induce regulatory T cells, and regulatory T cells will suppress the immune system from recognizing parasites, and this is how parasites can then thrive and survive, right? So, however, there's also conditions where regulatory T cells could be problematic for certain types of parasites. Again, that duality comes in. It depends on the type of parasites and what kind of environment it thrives. So much so that this concept here can even lead to paradoxical events. And now you can also see, and I'll tell you in a moment, but now you can see, for example, that look, if parasites induce these regulatory T cells that suppress the environment, then such same regulatory T cells could be helping cancer elsewhere. And perhaps this is how ivermectin or fenbendazole could be helpful for fighting cancers. Now, let's talk about the paradoxical component. It's known that certain parasites can, in fact, in certain contexts, help cancer growth, and in other contexts can help fight cancers. And again, this could very well be because of these regulatory T cells. So the last thing I want to tell you is this paper that this scientist provided for me. And this paper investigated, it was from 2017, and investigated that very concept. How do regulatory T cells and parasites help towards cancers? And this was done in mice, and what they did is they infected these mice with this specific type of parasite. And let's see if I'm going to remember the name. I think it was something like Trichuris murus. But again, I'm trying to say it because it's a funny name, but it's Latin. So I don't know if I said it correctly, but they infected these mice for a very long time, and then they wanted to see what happens. And indeed there was inflammation, very similar to what you would see in people with inflammatory bowel disease. So epithelial cells were growing more, and stacking more. Epithelial cells are the cells that line the gut. So line the intestines of these cells. They had abnormal crypts. These are glands that produce epithelial cells because they have to be replenished all the time inside our gut. So they were becoming wider. There was more invasion of immune cells into that lamina underneath the epithelial cells. All of this is very reminiscent of inflammatory bowel disease, but they also had neoplastic markers, meaning markers that are suggestive of future cancer development. So the way it goes is you have normal cells, then the cells divide more, but they're still normal, then they start to look abnormal, and then they lose control of the vision, and then you can create a tumor, and then tumor can be benign, but then it can also start invading tissues, and that's when you have cancer, and cancer can also become metastatic, where it can travel to other body parts, right? So that's kind of like the progression, and what they were seeing is that these crypts were also starting to becoming abnormal. So that's one of the earliest markers of what could be a potential future cancer inside the gut, inside the intestine. And they also saw that there was pre-adenomas forming. So what are adenomas? Adenomas are benign tumors inside our gut, but adenomas can eventually become cancerous, and that's how you get adenocarcinoma, I think, colon cancer. So that's what they were observing. So overall, these mice, because of this long parasitic infection, they started to have this neoplastic changes. And what's really interesting is that they also tested other bunch of mice with carcinogen. And the carcinogen led to similar changes as the actual parasite infection. And parasite infection might have even caused more neoplastic changes. Now, the next experiment is the key one for our story. They took mice that had a mutation that predisposed them to form these adenomas inside their gut, in their colon. And the colon, the large intestine, is where that parasite resides. And when they infected these mice with the parasite, they noticed that there was now more of these adenomas, but not in the colon where the parasite resided, but in a small intestine. So it shows you that the parasite, even though it's elsewhere, it happened these neoplastic changes to occur elsewhere. So it's a systemic problem, meaning it's a problem that they can help cancer development somewhere else in the body. And finally, here comes the regulatory T cells. They decided to target regulatory T cells because they knew regulatory T cells can contribute or help parasites thrive and survive. So that they decided to take them out of the equation by targeting them with antibodies that attack that CD25 marker on top of regulatory T cells. And they did exactly that. And when they did do that, they these mice, after infection, no longer had that increased amount of those adenomas. So it helped. Here's the kicker though. When they took healthy mice, normal mice that were never infected with parasite, and they did that same antibody treatment to target regulatory T cells, meaning they were healthy mice and they removed their regulatory T cells, those mice also had more adenomas afterwards, which shows you that duality role. In this case, it either the amount of regulatory T cells was important, so that healthy mice, if you remove the regulatory T cells, that could have been a problem because you didn't have enough, or their function change. And if you had parasites contributed to either too high amount of regulatory T cells or regulatory T cells that were slightly different, that were now helping cancer development. And by removing those regulatory T cells, you could potentially help to target, help these mice recover. And the same thing might very well be happening with ivermectin and fenbendazole, but we're not really investigating this. This is just a great example where these authors were showing, look, this regulatory T cells could play a role here. Okay, my stick battery died. So now I gotta do this by hand. So this is how ivermectin and fenbendazole could be helping fight cancers in a way besides their own direct effect on cancer development itself. It could also be an indirect effect via targeting parasites themselves. And by targeting parasites themselves, what they could be doing instead is helping reduce the regulatory T cells pool that could be helping cancer cells in elsewhere. And that could be another indirect mechanism how fenbendazole and ivermectin or parasite fighting drugs could be helping cancer, because as a consequence, they could be reducing this regulatory T cells that could be helping cancers elsewhere. All right, I'm gonna stop it right there. I was hoping I'm gonna make this video without stick dying. I knew it was an issue because my phone was not attached properly. But please check out my Patreon as well. My patrons get to vote what content makes it to YouTube, number one. Also, I am starting a new project on building a guide for healthy living. I want to make this guide freely available. Now, I have adopted a lot of changes in my life because of the pandemic in order to help myself, but I want to basically create a guide for everyone. And I want to ask help from community to help me do this. So I now have regular meetings on Patreon. So go to my Patreon. You can be a free subscriber in order to participate, and let's hear your wisdom. What are the best mechanisms you can think of that increase our health? And the reason why I'm interested in this is because, in my opinion, that is the most important way in how we got to protect our immune system and fight the consequences of the pandemic, aka the exposure to spike protein and its negative consequences. All right, I'm going to wrap it up right here, and I'll see you guys next time. Bye, everyone.